sutural cataract along with the ptosis and exotropia. So this is the clinical picture. You can see there's a faint lenticular opacity in the right eye, and the left eye had a sutural cataract. The corneal diameters of the left eye were small. Her keratometry was steep, and she had an asymmetric presentation with left more than right eye cataract with the microcornea as well as the microphthalmos. Father also gave history of intellectual disability and said both eye had low vision. When we examined him, he also had asymmetric microcornea along with nystagmus and had an absorbed membranous cataract for which he hadn't received any medical attention so far. We did a pedigree chart, and by what they gave history was a lot of males in 3 generations were affected with low vision. That is what the history told us. And when we screened the mother side, there was no such history of low vision or any other systemic intellectual complaints. The patient had low set ears, had some speech delay and intellectual disability. Along with that, a very short stature. So we were trying to put this all together with cataract, but we couldn't reach at a particular diagnosis. So we sent the patient for a pediatric consult. They suspected that it was maybe a Noonan and did a hearing and a detailed cardiac evaluation along with a renal evaluation, which came out to be normal. She had hemivertebra and butterfly vertebra and also some dental anomalies that the pediatrician noticed. Like you see the notch-shaped incisors in the child. So we again went in for a detailed dental consult, and you can see the child has low set ears and has these prominent central incisor teeth having these screwdriver kind of shape along with vertical notches, which is known as the Hutchinson incisors. And there were some incisors, central as well as lateral, that were missing, and the same findings were seen in the lower teeth as well. Radiographs depicted taurodontism. That is, they have the pulp of the teeth is shifted towards the apex and the roots become large. So these are again typical Hutchinson incisors. And when we put it together, sutural cataract along with dental anomalies, the first and the most common diagnosis that we need to know is the Nance Horan Syndrome. So it's also known as the cataract otodental syndrome, and it's an X-linked disorders. And world over, I could find about 70 cases that have been reported so far, described by Nance and Horan and Wilson simultaneously. So for ophthalmologists, congenital cataracts, which are sutural, along with microcornea, microphthalmos, and sometimes nystagmus, and characteristic dental anomalies, points towards the diagnosis of NHS, and they might go unrecognized because their other systemic features are very subtle. So the abnormalities are located on the NHS gene, which is located at the XP chromosome, and there have been a lot of deletions which have been and nonsense mutations which have been reported, mostly located in exon 1, 4, and 6 of the NHS gene. It has a role in cell migration, and hence these tight junctions, when get affected, patient has has cataract once affected. It's also known to be X-linked dominant. So if we look at our pedigree, we kind of asked again whether only males were affected. We haven't actually examined the affected that the females which were undergoing, which were passing on the affected gene were maybe because of X inactivation. So what is X inactivation? Maybe we have actually covered it once. Veronica taught us that in the late blastocyst stage, one of the X gets inactivated. So it can be either the parental X or can be the maternal X. So the female would be a mosaic, carrying some cells which might have one affected X, which is silenced, or the unaffected one. So that's why the females become carriers and also have lesser severer phenotypes than the males. So in cases of NHS, the male would have an intellectual disability, would have bilateral cataracts, they would present early and they would need surgery with the same findings, microcornea, microphthalmos, and permanent dentition abnormalities. In the females, they have the same gene affected in half of the cells maybe. So they have sutural cataracts, their vision is not usually affected. They do have facial dysmorphism, but which is milder as compared to the male, as well as dental anomalies. So our patient also had some additional findings like hemivertebra and butterfly vertebra along with vertebral flex. Yeah, which is an we ordered a clinical exome sequencing after debating whether we should do a microarray, and we found out that yes, there was a pathogenic mutation. It was at exon 7 of the NHS, and it was X-linked dominant again. Again, if we review literature, we could explain all findings: bilateral cataracts in the male and sutural cataracts in the female, along with mental retardation, as well as the dental features. What we couldn't explain were the skeletal abnormalities. And I think Krishna would cover here in the NGS, we actually could find a large deletion on the chromosome 16 as well, which is known to cause all these variable vertebral defects in the proband, in the child. We also confirmed the mutation by CMA and the standards of the father. The father also carried the same variant. And so our case had actually dual diagnosis because the cataracts and the dental features were because of the Nance Horan syndrome, because of the NHS abnormality, and the other segmental defects that we saw in the child were actually because of a chromosome 16 deletion, which wasn't there in the other family members. So if we look at the latest review on NHS, there are around 87 mutations, and they say that there's a lot of interfamilial variability that you can see in NHS, and sometimes frame shift mutations can lead to more severer ocular phenotypes. So we should do a detailed, comprehensive ophthalmic examination whenever we suspect a case of cataract, and that also running in families. A detailed pediatric evaluation also should be done after examining the parents dilated. Cataracts with microcornea, and where you are suspecting an X-linked inheritance, sometimes as ophthalmologists we forget, we should look for dental anomalies because the other systemic abnormalities might be very subtle, and they might be, we should remember that's an X-linked dominant and might have variable findings in the female and lesser severe phenotypes as compared to males. My acknowledgements to my genetic teams, my mentors at PGIMER. That's all. Thank you.
Thank you, Dr. Saveleen. That was a really good case. I actually have questions. Did you do the sequencing first or the microarray first?
We did the sequencing first because that's what we did. The pediatrician said that they couldn't explain the vertebral anomalies. Maybe we do CMA and then we kind of go for CS. Rather than that, we did the exome first and then we confirmed it by CMA.
And my second question is that in a case like this, so it happens here as well, where there's a multi-systemic affected child and we do one test and part of the phenotype can be explained but the other can't. So are there any signs that make you go that okay, maybe a chromosomal microarray will also yield a positive result in this case? Sometimes they could just be co-occurrences that can't be explained, but any particular signs that no, maybe a microarray will in this case explain more?
I don't know. I think you might tell me better. What I understand is if I wasn't sure that this was cataract and dental anomalies, I might have just done a microarray, right? But I couldn't. The rest of the findings were kind of not fitting, so that's why we went for exome. You tell me, what's your take on this?
I too would have, after doing if there was no immediate diagnosis after running it through like you were doing it on OMIM, I'd have used a couple of more softwares to get my differentials, and then based on whether it is a monogenic disorder or what kind of mutations are seen more often in it, come to a decision which in terms of cascade testing, because that's usually how we all do it here. But yes, I was wondering myself that if this case would have come to me, would I have stopped at exome and said, okay, this child has Nance Horan, but I really don't know why she has the skeletal abnormalities, or gone further. So it's quite good. And also, I'm so sorry, I don't know what chromosome NHS gene is on, is it on the same one in which you got the microdeletion, or is it a separate region?
It's separate. It's we got
Separate.
the deletion on chromosome
Right.
16, and this is X and P.
Oh yes.
Yeah.
Oh
So
yes.
it's
XX. Yes.
yeah,
Yes. Yes. This is X.
it's
Yes.
totally different.
Different.
And
Right.
chromosome 16 was
Oh,
also, we were
interesting.
lucky we got a deletion which had known phenotypes.
Yes.
So it actually fitted. But then again, if NGS would not have picked up this deletion, I don't know, every time it doesn't.
It does.
So so we were lucky that way also, because otherwise also, if we had just got the NHS in NGS, what would we have done next? We again, the deletion, we would have needed another test, right? That's what I understand.
Right. Any questions or comments from the people who have joined in?
Okay, I can see Hannah joined in today.
Yeah.
Thank you, Hannah, for joining. Are you there?
Hi. Yes, I'm here. I just no video because I'm in transit to work this morning.
Yeah.
I'm glad to be here. Since you called me out, I actually do have a question. Did you have the opportunity to examine any of the other females in the paternal family?
I couldn't do that. We tried a lot. We just examined the family of the mother. We could actually examine. The father's family we couldn't. We just went by history. We couldn't examine the females in the the father's family.
Of course, and it's such an. Oh, go ahead.
No, go ahead.
I was just gonna say it's when someone does have the opportunity to examine females, let's say in your situation, father was unaffected. It's such an important carrier sign in the females to have. It could be a prominent Y suture or it could be a proper opacity. It can be both. It's such an important sign, and it's there. I don't know what the earliest report is, but unlike some other excellent carrier signs that appear later, like after puberty, this one is there. It can be, we have a 2 or 3 year old who's known to be a carrier who has a Y sutural cataract. So if your clinic allows, if the families are able, or if you're seeing a patient who doesn't have a family history, taking a look at parents, of course, might give you these clues.
Yeah, we could just do the parents because we couldn't do the other, because that's what we were wondering. They were just giving history that the males are affected. We haven't really examined. It's just by, retrospectively are thinking that, okay, these females must have been carriers of the.
Right. Yeah, the father was the big clue for you.
Yes. That's the scenario in our country. Now he would never seek medical attention. At his age, he wasn't operated for cataracts. He just had them, and he had a membranous kind. We couldn't pick up I don't know, Hannah, what do you think? The males do not have suture, right? Whatever I have read, it's that they have a total cataract.
Yeah, that's most typical, is the total cataract. But they can have the sutural. So, but sometimes it comes down to, as you mentioned, the mutational differences. So a frame shift leading to a more severe phenotype. If you have different mutations that have less loss of function impact, you could get a sutural. We have a case where in the process of publishing of a male with a Y sutural in one lens and very asymmetric. He's 6-7 years old and only needed surgery in one eye so far. So it can definitely come down to the mutational difference, but your classic cataract is going to be that total white at birth in both eyes with microcornea. But yours is important too, to publish what it looks like without surgery. Something to think about, because we only think about, typically when you went on OMIM and you look in the case reports, you're thinking about the bilateral congenital cataract phenotype, but having an adult without surgery, knowing what the cataract looks like later is important to know, because I don't think many people are aware of that.
Yeah, we see lots of those in us.
Most of the childhood cataracts, if you leave, they'll turn into a membranous cataract.
Membranous cataract, yeah.
Lot of them.
Yeah. We don't know whether it was sutural to begin with. That's why I'm asking, because most of the reports, like you said, are for people undergoing surgery. That's how they present, right? So they present with a sutural or a bilateral, white cataract. Now this one has been long standing forever. So anyways would present as a membranous. I think whatever it was to begin with.
Unlikely to have been Y sutural, more likely to be total white.
Well, yeah, that agreed. What was the brother's finding?
The brother is absolutely normal.
Oh, and how is that possible?
Because it's the father's, I mean, yeah, you tell me,
Yeah,
that's what we were,
it's possible. Yeah, it's possible. Father would give the Y.
father
Mother,
would give the Y to the, yeah, so the
yeah, yeah.
X is
I
from
was
the
getting
mother. Yeah,
yeah,
from the, yeah, the son,
sorry,
that he
sorry.
can be normal. You confuse me. The son can be normal, yes.
Great, yeah. So, yeah, so brother will get, yeah, so brother
Yeah,
more chance of
now
getting normal.
this child's, 50% chances that her sons would get affected, right?
Son
The
would be
proband,
can
yeah.
be affected. Yeah, agreed, agreed. I think Deepak has raised his hand. Deepak, please go ahead.
Yeah, hi, thanks. A great case. So I was just wondering, from the literature, is there anything about the chromosome 16 variant that you co-found in this case? Like, is there any predisposing factors for that particular variant? Or if we had screened the parents, would we have picked something up?
We did screen the parents. We couldn't
Mhm.
find the same. We just found the NHS in the father.
Right.
And literature has reported chromosome 16, this deletion particularly responsible for these defects, but not both of them together.
I see. So is this deletion a de novo occurrence, or is it known to
That's
occur?
what we think. That's what we
Okay,
think. As of now, it's a de novo deletion that occurred.
I see. Thank you.
Any other questions or comments from
How
other?
frequently, Sheza, have you encountered NHS? Or maybe Hannah has a lot, because
I haven't
she's
seen even a single case of NHS so far. It's that rare. Yeah, read about it. As Hannah was mentioning, often we take the example of, carrier screening. In them, NHS and mothers of children where, xanthomatosis, like cerebrotendinous xanthomatosis. In those, these mothers, they are carriers and they have cataracts. So to look for cataracts when you see males with cataract, bilateral cataract in males with some systemic abnormalities. So in those examples, I've always read about NHS, but hardly ever seen in so far.
Yes.
It's quite rare. As you mentioned, hardly 70 cases all over the world. So
It is, but then, I was talking to Dr. Karthikeyan, and he said that it's underreported, because how many times you check for teeth, everything, maybe, that's what I started doing. So every microcornea and, cataract, I, start asking them to open their teeth and see, because maybe we are missing them.
Agreed.
Yeah.
It could have been missing them. Agreed. So a case is very interesting. We often come across these kind of cases where the entire spectrum of findings doesn't fit into one particular disease, and you keep scratching your brain trying to fit everything into one syndrome. And often you tend to not find any kind of correlation or any kind of syndrome where it fits into. So probably then you have to think about two things at once, and can be possible, right? To have one particular, like you showed that the vertebral anomalies were not fitting into the classic NHS, and it was something else which came up. That's how genetic diagnosis can also help us, solve this kind of puzzle.
From the micro deletion that we got, is there anything that puts her on a stronger surveillance, or any like risk factors that for any other organ systems that she needs to now go on?
Luckily, no. We had screened beforehand only, but just that again, it's de novo. So, besides her son's having cataract, and we advised an early screening for cataracts, for the next generation. So we didn't,
Okay.
She doesn't have any systemic problem.
Okay.
So where, like, that you can.
Sorry, I couldn't hear you. I think your signal's cracking a little bit.
Yeah, sorry. Yeah, I'm coming back. Yeah. Can you hear me now? Sorry.
Yeah, yeah.
So I'm coming back to your first question, as when you have, a lot of systems being affected, what will be your first line of test, genetic test that you will order?
Like I said, so I would like to have some sort of a suspicion, at least a provisional diagnosis in terms of what system it is. So mostly, even if that needs me to not give the patient an answer in case I don't have one immediately in my head, I will call them back. I will do my due research before, because mostly that unless you have affording patients, that maybe one test is where they'll stop and they'll not go for a cascade testing kind of thing. So once I have that, I look at what kind of mutations are most reported. So like an Angelman would straight away we know would need like a del-dup imprinting disorder, whereas a Nance Horan, for example, could have point mutations in which then we'll decide whether we do NHS or. But because it's a diagnosis that does not come to you firsthand as soon as you look at a patient, I will still order a broader panel test so that it covers genes that maybe or other diagnoses that I have not thought about, and proceed in that manner. But but I'm trying to think whether I think if Hannah could comment on this, that if part of your presentation remains unanswered, for example, the skeletal abnormalities in this child, how soon would you think that, okay, maybe she needs a microarray or del-dup analysis?
Yeah, so our approach typically is we do certainly the same as you, coming up with that differential, seeing what might be most likely. And we, instead of exome, it's difficult to get through insurance companies in the US. We do curated panels. So like for this patient, I would have selected, we have a cataract panel that we customize, and we do run it on an exome backbone. So we have, capabilities to do more when we'd like. If I couldn't put everything together as in this case with the vertebral anomalies, then I would have considered either a reflex to whole exome, because that's just our pathway, or a microarray. Probably during the results session, I usually take that opportunity, depending on how the family is coping with the diagnosis, to discuss, should we do more testing?
Right. Yeah, similar. Thank you. Thanks. So if you don't have any more questions, we'll move to the next discussion for today.
I think, is it my connection, or was it lost for everybody?
In the middle, it was a bit, but now it's fine.
Okay.
Yeah.
Okay, fine. I'll ask you later what Hannah said. Maybe we'll move on to the next.
Yeah. Yes. Dr. Krishna, it's the floor is yours.
Thank you, Ria. And it's a great presentation, Selvin. And these kind of interactions definitely provoke us to look more into the ocular things. And hopefully, these kind of the webinars and what you are conducting definitely help us to make a better establishment of knowledge. So let me share the screen. I think my screen is visible.
Yes, right. We can see it. Yeah.
Good evening, everybody. I'm Dr. Krishnasai Reddy. Today we'll learn about next generation sequencing, as it is a groundbreaking technology that we are using. So I'm just making it very simplified as much as possible. So it's a revolutionizing genomics with cutting edge technology. Yeah. So today we'll discuss about the introduction of the brief history which related to the NGS and a little bit of Human Genome Project, and we'll see what is NGS and how it works, the workflow of NGS. Soon after, what are the clinical applications, what we are looking at, and the challenges. The complexity in data analysis and variant interpretations are the biggest challenges in NGS. So we'll have a look on it, and then we'll discuss the future directions and conclusions. This is the thing what we can simplify this. Yes, the next generation is in our hands. This revolutionizing technology allow us to approach in a new way to the genetic disorders. In prenatal, through pre-implantation genetic diagnosis, we can detect early and we can prevent it. We are not diagnosing any genetic disorders. We are not focusing on the diagnosis, but we can prevent it to the next generation. So it also decrease the inherited disorders, as Alvin said, as we have come across the X-linked recessive things. So we can predict those things. We can definitely have an option where we can prevent to the next generation through early screenings and early NGS platforms. So this particular revolutionary technique can change the spectrum in genetic testing. Before going to that, what is sequencing? We have to know that this is a next generation sequencing. What is sequencing? Just come out of the box here. Just see the picture. It's just a 2-second video, or 2 to 3 second video. A boy is jumping. So the boy is jumping is just an action. It's just representing an action. So whenever we have to oversee it, we have to make a frame by frame through a sequence that is called 24 traps. We can see 24 frames. So each frame indicate an individual pose. We collaboratively make a sequence of those poses will give a particular action. The boy is jumping. That is what a video can make. So a sequence of pictures give a data of, an action. As such, we have a DNA structure. As we know that we have nucleotides, the basic building blocks, adenine, thymine, cytosine, guanine. They have already, a huge data which has been sequenced in an order. Each sequence, each phase of the sequence has individual effect, individual phenotype. So we have 26 alphabets in our English, as I mentioned there. CAR, the group of 3 letters, CAR indicates car. We just replaced R with a T. CAT, instead of R, there is a T. It indicates cat. So just it's normal and universal phenomena. It's not about just NGS or just any technical thing. It's just universal phenomena where a single change can lead to a different character, different subject. As we have the code of life has a single base pair change can lead to different phenotypes. So we have a human genome project. I think all from the same background, we know that. So this particular human genome project is an outstanding achievement that human species have ever and will ever accomplish. Because reading this code helps us understanding how we are made, how we are all related, and how errors or mutation in our DNA cause disease, and how we respond to best to the medicines. So all these things has been come into an end where we can just take it as a reference through a human genome project. So whenever we can see the timelines, Frederick Sanger, the Sanger sequencing is a golden turn of the sequencing establishment, where first genome has been sequenced, that is out of 5 kb in 1977. And then in 1990, the first human genome project has been initiated. Then in 2003, the human genome project completed, but with gaps. Then in 2005, the phenomenal thing called next generation sequencing. Soon after Sanger sequencing, there are a lot of first generation, second generation, third generation sequencing. Then it upgraded to next generation sequencing in 2005. And then in 2022, March 31st, we have a complete human genome project they have complete filled the remaining gaps. So these are the basic timelines we have to be acknowledge. Yeah. Here now we can go with NGS and its workflow. We know what is sequence, and you know what are the basic nucleotides what we have in the DNA. But what this sequence, what this next generation sequencing is a massive parallel sequencing technology that offers ultimate throughput, scalability, and speed. It's a groundbreaking advancement. As you can see, the puzzle shapes what I mentioned there. An individual puzzle chip has an individual sequence. Whenever we can combine whole chips together, whole the, the papers together, we will get a whole picture, a complete picture. That is what NGS, the baseline mechanism of NGS2. So this unprecedented speed and cost efficacy technology will definitely change the complete scenario of detecting genetic disorders. The future is now, as you can see, Ion Gen Studio S5 system and Ion Torrent Genex systems. These are the overlook of the NGS missions what actually available in the market. I'll go deep. Every NGS has the basic phenomena and basic format and the basic workflow. There are 4 steps: extraction, library preparation, sequencing, and analysis. Let me explain with a 2 minutes video here. I think this animation video can definitely explain you better. So first we'll get the sample. The sample has been extracted by the laboratory scientist, and then we have extracted the DNA from the cells. Then those things has to be sent to library preparation. So this library preparation is nothing but making the whole fragment of the DNA, the whole DNA into fragments. Each fragment is attached with the adapters. Adapters are nothing but oligonucleotides. Those oligonucleotides has a sequence which have complementary, and then those fragments have been denaturated, and denaturated fragments has been attached with the adapters, and we have a workflow chip. The chip already have an adapter which has been sequenced with the complementary base pairs. So this particular fragment, it's a single fragment. It has a polymerase chain reaction again. It has been sequenced again with the PCR, and then we have washed it, and then we removed the total thing. And next we have a bridge amplification. I think it has been, wait, maybe it's little fast. I'll explain. Maybe confused. Yeah. See, those are adapters, oligonucleotides. Those adapters has to be binded ligand to the both the ends of the fragment. Each fragment has 2 adapters. Each adapter has a sequence that is complementary to the sequence which already placed in that workflow chip. Every sequence mission has a chip. That chip has already sequenced adapters. So we have already, it's kind of a magnet, both magnets. So this magnet we have attached, adding to the fragments of DNA what we have acquired, and we soon after the adapter has been done, we are sending to the the chip. 1 minute. I'm sorry. Yeah. It's a complex thing actually to learn. Yeah, these particular adapters has been fixed to the workflow chip. So soon after the washing, the PCR has been get the complementary base strip, but that complementary base strip is not attached to the workflow. So that's why it removed soon after washing. Then the bridge amplification started because the both ends has stick to the work strip. Then again PCR has created the complementary strand, and then again a multiplexing bridges has been made throughout the chip. This phenomenon happened to all the fragments in the DNA. Then the primer added to the fragment, that is oligonucleotide. Then it actually picked the nucleotides which have already fluorescence in it. And ELISA can detect what are the sequent element, that is T, A, C, G. So whole sequence has to be recorded through a illuminating ELISA technique. So this sequence has been for the millions of copies simultaneously did by the next generation sequencing mission. So that is how it works actually. So soon after this, we have a sequence. So we did the same process for millions of fragments of DNA. So we get all the sequential data, what we have get from the ELISA thing. Then we have a long reads and short reads. So those long reads and short reads has to be processed. So the process has been initiated through a TXT format. It's a bioinformatic tool, and the analysis has been cope up with the informatic softwares and all. So these bioinformatic pipelines can take the reference sequence from the whole genome project, and they got the whole data from our genome as a reads. So we have comparing with the reference sequence, and ultimately we have sequencing the whole genome. And then we cross-checking with whether there is a change in the particular, the particular fragment or the particular location of the gene, and it leads to the variant. So those variant is called VCF, variant calling format. It's a bioinformatic tool where we can get all the variant details. So we can get like it is pathogenic or likely pathogenic, benign, or the software it's analyzes that, and then ultimately the genomic report has been established, and we have discussed with the clinical geneticist or a genetic counselor. So this is how actually it works. So there is 4 steps. The 4 steps initiating through the differentiating process. As I mentioned, this is DNA extraction, library preparation, sequencing, and cluster growth. And this is the step 4 of analysis. So let me quickly brief it. So this particular DNA extraction might be any sample, that is cell culture, fresh frozen tissues, or FFPE, formalin fixed paraffin embedded blocks tissues, whether from the tumor site of tumor, or like for hematology, it's a bone marrow sample, whatever it might be. So we'll get the sample and begins to with the collection of biological material preservation to prevent degradation as regular as the format of what we are following in the laboratory of DNA extraction methods. Then the library preparation. The library preparation is nothing but getting the adapters. Adapters nothing but oligonucleotides, which actually bind to both the end of the fragments, which ultimately leads to hybridization to the workflow cell. So this is how after adapter ligation, the library undergoes several crucial steps, including PCR amplification to enrich the sequence of interest, size selection, and remove unwanted fragments, as you have seen there. And there we have a deep analysis of quality control to assess library quality and quantity. So those library preparation, what we have seen, it's different from method to method, whether we are doing for whole exome sequencing, or whether we are doing for clinical exome sequencing, or we are doing for a whole genome sequencing, or we are focusingly targeted on particular panel. So this library preparation is a must, and it has a deep classifications. So it actually allows to do a better. Yeah. So
sorry,
then
Dr. Krishna,
we'll
just
get
trying
a
to mute
sequence.
the. Yeah.
Okay. Can I continue?
Yes, please.
Yeah. Then we'll go with the sequencing, and we have get the whole process of clonal amplification and sequence by synthesis, as these are ultimately get the image capture and base calling. These are the steps what we have gone through in the sequencing mission. Ultimately we'll get FASTA files and lead to variant calling format, which have the sequence of the reads. So this ultimate variant calling format will give us the annotated file where we can have the variant classifications of whether this particular variant is, it's a pathogenic or likely pathogenic or benign, or VUS. So this is what a huge process, but of course it's a complex process in interlinked with the different software proclivities. But in a superficial way, this is how NGS works. But as I mentioned, in genetic diagnostics, variant classification forms the basis of clinical judgment. Despite the international guidelines and principles, there is still significant inter-laboratory variation in classification of genetic variants. So the only problem with these, the wrong classification can misguide the clinical judgment. Whenever you are getting a variant, but you are just, giving a likely pathogenic variant as a likely benign variant, then definitely it might be, error, red flag in the diagnostic spectrum. So this is what a clinical judgment in patient care, including several consequences in risk stratification among family members. As I mentioned, we have a methods of NGS. We have whole exome sequencing, whole genome sequencing, clinical exome sequencing, and we have a dedicated target panels. And what are these? As you can see here, I think everybody knows that we have a whole genome, and that particular slice of an apple is called exome. What we are seeing coding regions of the whole DNA thing. So we are focusing on exons. Whenever we are focusing on exons, then we can go with whole exome sequencing. Or whenever we need to go through the whole genome sequencing, then we need to go through a whole genome sequencing test. So there are three to four methods where we can focus on. But why? Why instead of gene panel sequencing, why can't we go with the whole genome sequencing for everybody? Why whole exome and why clinical exome and why targeted? Because this particular gene panel sequencing involves analyzing a specific set of genes. So this panel consists of a particular thing, whether you can go with the cardiology panel, nephrology panel, neurology panel. So this particular panel consists of a set of genes, which are from 120 to 200 set of genes, which has a deep depth. I'll explain what is a depth in NGS. That is much more essential to evaluate the report whenever you're getting. So my whole session, my whole thing was to explain you, was to make you aware of what is depth. Whenever you are getting an NGS report, then definitely you must ask the provider. So what is depth? So that is what I want to mention in the next slides. So this particular gene panel is essential. Why? Because these panels are designed to target genes that are known to relevant and specific clinical questions or disease under investigation. So whenever we are focusing on that particular panel, it has a more chances to get an accurate result rather than clinical exome and whole exome. So this panel may include a few genes from hundreds of genes depending on the purpose of the test. Whenever it comes to clinical exome sequencing, covers an entire protein. We mention it as whether it is a clinical or whole exome sequencing. We focus on the protein coding regions of an individual genome known as an exome. The exome comprises approximately 1 to 2% of entire genome, but includes a vast majority of disease-causing genetic variants. So this is what we are focusing just on exome. It gives a sustainable result, but not as depth as the targeted panel. And whole genome sequencing, it also gives, the entire genome has been sequenced with a huge data, but it also can't give the essential depth which has been provided by the targeted panels. So this diagnostic rate is highest among the all genetic tests. The whole genome sequencing has a highest diagnostic rate. We can, because every intronic region has been sequenced and we can see the very minute SNPs and all. But even though inwards, we can have a better depth in targeted panel sequencing. So the application of whole genome sequencing as a first tier analysis will extend gene analysis coverage to non-coding regions, as I mentioned, thereby reducing unnecessary repetitive testing. Further, this method allows the detection of variations such as CV and mitochondrial DNA, copy number variants and mitochondrial DNA, which cannot be evaluated using whole exome sequencing because it's a coding region. I mentioned it earlier, targeted sequencing, it has panel of 20 to 200 predefined genes. Those genes might not covered in whole genome sequencing, whole exome sequencing, because targeted sequencing, targeted panels has been designed with a unique library. So this particular library has been, they have designed to the up-to-date information. They can, refer the recent articles, recent review literatures, and the whole new research statements and the guidelines. So majority of genome analysts and the clinical geneticists may impact together to find a different platforms. That's why whenever you are giving a particular panel test to the companies, you can see every different companies have their own set of genes for the panels. That is how unique they are. So majority of us don't know. We think those are common panels for every company, what they are provided for cardiology panel, nephrology, neurology panel. But you can definitely see slight changes or a better gene additions, better number of genes additions to the different panels. So that is how it works. So you can see the depth. You can see the depth of the reads. So this particular targeted sequencing has a more depth when compared to the whole exome sequencing and when compared to the whole genome sequencing. So what is depth of coverage? As I mentioned, the average number of times each base of target region has been sequenced. For instance, a depth of 30x means on an average, each nucleotide is sequenced to 30, is sequenced and reads 30 times. So what we expect from the companies, they have to do, they must have to do 100x. Inka matlab means they have to sequence the target region for 100 times. So, but you know, the basic guidelines is about typically 30x is considered, 30 times is considered a standard for human genomes. But majority of companies will provide us an assurance we are sequencing 100x. So that is how the cost been reduced. But majority times they aren't doing the sequencing for 100x, but they are doing 20x, 25x, or 50x. But that is what we have to be much focused on to get a good depth and good result and good accuracy of the test. And the importance of depth, what is the importance? Then we have an accuracy, that is what I mentioned, accuracy, variant detection, and confidence on the report. So this particular depth provides us the accuracy of, you know, a small random errors affecting in the final sequencing, and you know, SNP SNPs, we can see the small insertions, deletions, and the SNPs through this variant detection. So the more depth you can find, the more simpler, I mean, minute changes from the base pair change. And the recommended depths, as I mentioned, whole genome sequencing 30x is recommended, whole exome sequencing 100x is recommended, because we are focusing on the exome, the protein coded region. That's why it need a 100x. For the targeted region, 500x. You can see there, 500x or more, because that depth is essential to figure out that particular thing. That's what targeted sequencing, or the panels are much more costlier than whole exome and whole genome. So balancing depth and cost, that is what we have to make to the, you know, patients, to the clinician compatibility. Higher depth increases sequencing cost and data analysis complexity. Therefore, a balancing between between achieving sufficient depth of reliable results and managing cost and data processing. So we can't able to, you know, offer the people, everybody of going through the panels, but we can choose and we can situate when it is really required, and definitely it is the only way to do. So then we have to go with the panels. And clinical diagnostics, high and consistent depth is critical for ensuring accurate and reliable diagnosis, especially in genetic testing and personalized medicine. And as I mentioned, the calculation, how it 100 base pair region is sequenced at 10,000 reads. So 10,000 reads by 100 base is 100x. I just illustrated with, you know, the depth where you can go to the deeper sea level of the sea, then you can get the treasure as such. So we'll get the results accurately when you have a good depth. That is what I want to convey you people. So these are the missionaries what available in the market now. You know, mini-sec system, MySec series, NextSec series, HiSec series, HiSec X series, NovaSec. So these are missionaries of NGS missionaries which have a different work and different workflow, and you know, different upgradations with compared to the market competitors. So as I mentioned, MySec, NovaSec, and NextSec are the popular next generation sequencing missions developed by Illumina, a leading company in genomics industry. These missions differ mainly in the throughput and applications what they have been going through, as MiniSec and MySec offers low throughput, making it suitable for small scale projects like targeted sequencing and microbial genomics. HiSec and HiSec 4000 are capable of generating large volume of data, making them suitable for whole genome sequencing. So I think you will get clarity that whenever we want to go for a huge data and deeper depth, so we have to go with a HiSec, then MiniSec. So that is what how we can differentiate the missions. We have used the different missions. And the next sequencing providers mid-range throughput, ideal for exome sequencing, and you know, NovaSec, and delivers a high throughput, enabling large-scale projects such as whole genome sequencing and population genomics. When competitor to the Illumina, Thermo Fisher Scientific, which manufacturing Ion Torrent series, and PacBio, Pacific Biosense, these are the missionaries which are available. These are advanced missionaries, and you know, PacBio is much more advanced than any missionary in the current mode, and known for its long-read sequencing platforms. Each company's platforms offer a unique advantage in read length, accuracy, and throughput to cater diverse reach and clinical needs. Yeah, so these are the sequencing missions and how the workflow and what is depth and what we have to focus on when we're reading the report, and what we have to ask the genome analyst or who is the NGS expert who are doing. So we must aware on that. So what is the depth and what is the the sequencing read length, and that is what we can ask. And you can definitely ask what is the mission, even a lot of systems and what, because we'll get to know that even the genome analyst is conscious that the clinician has better knowledge on these kind of things, so we have to give the better result, then they can go with the better clinical indications to explain to the patients. And as we know, cancer genome, the applications which have a wide range of applications. In the first place, we can see cancer genomics. As we know that cancer is a genetic disease, irrespective of the type of mutation. Mutation can lead to, every cell has to be grown at a certain pace. When it has been mutated, the oncogene, the proto-oncogene converted into oncogene, then leads to aggressive mode of cell division and lead to form a tumor, the tumor lead to be traveled to metastasize to the other parts of the body, leads to cancer. So these are the things which has to be focused, because these are like life-threatening conditions where we have to focus, and the ratios has been significantly increasing day by day. So NGS has been a brand new technology that can definitely give an answer for this. As I mentioned, proto-oncogenes usually make cell to grow and divide in a certain pace, but when it has been mutated, then leads to oncogene. As we know that cancer has been rise through inherited mutation and acquired, that is called germline and somatic. We know that how it has been inherited from generations to generation, that is called hereditary mutations, like we can breast cancer, cervical cancer, not cervical, colon cancers. These are the cancers which are acquired from the families. And somatic mutations, hematological cancers and bone cancers, which are somatic mutations which acquire through epigenetic factors, or it's a random mutations. So these cancers has been significantly detected by the NGS. NGS identifies somatic mutations in cancer, especially include single nucleotide variants, SNVs, insertion, deletions, and structural variations, which can help in understanding the molecular basis of cancers. So mutation detection and copy number variations and tumor heterogeneity. So by sequencing the multiple regions of the tumor or multiple tumors from the same patient, NGS can reveal the genetic diversity within a tumor, which has implications for treatment resistance and disease progression. So tumor heterogeneity is definitely one of the most prominent thing what we can do with the NGS thing. And recently I think there is a lot of things have been going on for the liquid biopsies, but the advancement and the upgradation has been making in NGS that we can capture from the circulating tumor DNA, providing a non-invasive method to detect and monitoring cancer earlier to the things. And recently I've seen an interview from Dubai. They are claiming we can even detect prior to the tumor formation. Might be that's one of the unique challenge that we are facing now. If that works, then it definitely make a huge impact on the cancer prediction early to the tumor. And then we know personalized medicine, in market we have a phenomena called try and try until it fits. So one size can't fit for all, no, even for the dress or the medication, whatever it might be. So we have to focus on this. There is no right prescription mode that actually available in the market, because everybody individual are different, everybody individual genetic makeup is different, everybody individual metabolism and pharmacokinetics and dynamics are different. Then how can we prescribe the same antibiotics to everybody? That is how the path-breaking thing NGS has to be segregated in the pharmacogenomics. This can definitely break this practice of try and try until it fits. So this is just a random reference. Factors involved in inter-individual variation in drug response. In a drug response, environmental factor causes 18%. It has an impact of 18%, and BMI, age, gender impact is 20%. But genetics impact of drug response with the body is 30%, and efficacy is 20%, and placebo is 5%. And 1 out of 3 people definitely will suffering with adverse drug reactions. Adverse drug reaction is nothing but whenever we are having a drug, so that drug will definitely suitable for us, but in some instance our metabolism, 1 minute, sir. Yeah, our metabolism, in some instance our metabolism can't able to match with our enzymes, the proteins what coded by the DNA, that leads to interaction between the drug to the body. So that is what drug interactions, adverse drug reactions, and drug to drug interactions has been depend on the genetic makeup of our body. So men are 50%, men are affected with 50%, and women are more likely 80%, genetically affected by the drug response with variation to the genetics. As you see here, we have given 3 drugs, 3 same pills to the 3 people simultaneously, but one A got 50% effect from the drug, and B got 100% effect, and C got 0% effect. That is what. And the studies are showing that pharmacogenomic testing, PGX testing, to do the huge, large population, it indicates, so a group of population can treat with the normal drug and dose, but majority it indicates with a different alternative drug, or increase or decrease in the dose. So that is why it's very, very essential for what I mean to explain, what I mean to put you to. So NGS is definitely essential in precision medicine. It's not such easy to make sure that every drug is simultaneously act on everybody, but this polymorphism, this change definitely to be ruled out, and we have to be classified individual, as we have an individual QR code for phone pay, Google Pay and all. As such, everybody need individual their genetic medical data, so there they can definitely get a better medicine, personalized medicine for them. So we know the polymorphism, SNPs, indeletions, variant number, tandem repeats, and restriction, fragment length polymorphisms. So these and all actually plays an important role in drug metabolism and pharmacokinetics and pharmacodynamics, how body react to the drug and drug react to the body. That is what. And we have a cytochrome P450, aldehyde oxidase, and sulfotransferases. These are the biggest enzymes which are responsible for metabolizing the drugs in the body, and those enzymes which are capable of producing through the protein, it's coded by the DNA. So that is how we have to figure out what are the enzymes that are suitable, what are the enzyme, how that particular enzyme metabolizing the drugs. So we have to focus on that. And our final goal is to maximize drug efficacy, minimizing drug toxicity, shift the goal of medicine from reaction to prevention, and aid in new drug development. As we know that gene therapies are being emerging through this particular things. So yeah, these are the applications, in-depth reviews, customized prescriptions. As I mentioned, it's customized prescription for individual to individual, and our main motto: decrease adverse drug reactions and identify your hereditary risk. As the risk of developing hereditary diseases, this will open a door to many different actionable plans that could improve your future health. And as I mentioned, gene therapies are emerging in a lot of variants, ways that actually gives standard protocols for us in a near future. And not only for that, we can go with agri-genomics, they transform the breeding, pest and disease research, and genomic engineering, cultivating a better kind of quality food, and better in mean of desirable traits what we have desired for the market dynamics, and genetic markers linked with the desirable traits informing cultivation, breeding decisions. And for every challenge we have a solution with NGS, to my knowledge, to my what I've made clear with that. So NGS can definitely a path-breaking thing that definitely change the scenarios. And as I mentioned, next generation in our hands, because we are not just diagnosing things, but we can prevent things through a pre-implantation genetic diagnosis, or the NIPTs, or the fundamental prenatal studies where we can see things, and couple carrier screenings where we can find they are carrier for the things. So we have a better strategy and plan for the next pregnancies, for a healthier pregnancies. So definitely NGS will give a solution for those things and enables a detailed analysis for this too. And we know that rare disorders, 6,000 to 8,000 rare disorders exist in medical literature. Regularly they are upgrading to extent now 6,000 to 8,000 rare disorders has been indicated in India. And 450 rare disorders, I'm sorry, not in India, it's worldwide. 450 rare disorders have been recorded in India, and 80% of rare diseases are genetic in origin. So almost 80% are genetic in origin. And actually, it's a slight mistake here. We have between 72 to 96 million burden. It's not people, millions of burden. That means cost effective. It's cost effective for treating the rare disorder children and making the treatment protocols. And obviously, there are no treatment strategies or protocols for the rare disorders. So the countrywide 72 to 96 million burden has been imposed. And our country also, they are focusing on to establish a more dedicated protocols for the rare disease treatment and management actions. So this NGS enhance infectious disease management by identifying pathogens and antimicrobial resistance to in prenatal and neonatal care. It screens for genetic disorders early. Additionally, NGS supports cardiovascular, neurological, and autoimmune disease diagnostics and treatment, improves transplant donor matching, and aid in public health by tracking diseases, outbreaks, and even genetic epidemiology. So as they have mentioned, tracking disease outbreaks. So we can definitely track, the COVID is an outbreak. So we can definitely track through the NGS, we can sequence what the new virus has been evolving, how that can transform into genotype, then they can make changes in our human body. So we can definitely have a sequence thing that we can definitely have a clue, a key for to proceed forward. And challenges, the main challenges is data management. Dealing with the enormous volume of data generated by NGS can be a significant challenge. Storing, processing, and managing terabytes of sequencing data can strain computational resources and infrastructure. One whole genome sequencing data will go beyond 100 GB. So 100 GB in times of data. So that is what a big thing. Data management is one of the challenges that we are facing currently. So NGS can be significant challenge. And cost, probably every individual has a cost problem. While the cost of NGS has decreased over time, but it still can be expensive for large scale projects, especially when considering sample preparation, sequencing, and data analysis. So even still, it has been very costly for the normal range of people, not only in India, from across the globe. And ethical and legal concerns, the generation of large scale genetic data raises concerns about privacy, data security, and ethical use of information. So what the governments and the policymakers has to be established a dedicated, kind of a good platform for the ethical and legal concerns for this. So they might be popped up soon. So we are hopefully to engage with the new systems and guidelines for the ethical and legal guidelines for that. And limitations, yes, NGS also has limitations. Detecting rare genetic variants in the population can be challenging due to depth of sequencing required and the potential for false positives. Sometimes we'll get some results that might be false positives. So we must aware of that, because the sequencing depth might not matched with the sequencing mission. And sample contamination, that would be a limitation for every kind of test. But for NGS, it should be very, very, very precise what we are getting, a quality and quantity of the data. And cross contamination between samples can occur in erroneous results, because we are focusing on the DNA. And non-coding regions, NGS is biased towards sequencing coding regions, leaving many non-coding regions because they are unexplored. And epigenetic information, NGS primarily provides sequence data. They just focus and they just give the sequence data bus. So they can't get more than that. So that's why methylation changes. We know that methylation changes will give a different phenotypic effect without changing the sequence. That is what the biggest challenge in NGS. That's why for Angelman syndrome, Prader-Willi syndrome, we'll go methylation specific MLPA instead of going through any clinical exam or whole exam. We are going through methylation specific MLPA to detect the particular conditions, because NGS can't detect, because there is no change in the single base pair, no change in the sequence. So definitely it can't predict. So that is what an epigenetic information, the methylation changes has been a biggest limitation in NGS. So that has to be improvised. Recently, I have seen Illumina as a recent advancement in NGS. They are mentioning we are now upcoming research advancement, we can cross check with methylation too. So that's where we are now. And future directions of NGS. So we'll see that accuracy and resolution has been improved. That what we are hoping for. So, and cost reduction, affordability, everybody has to be take, because the only thing is awareness should be maximized. Everybody should aware of what we are going through, and NGS should be a great platform where we can get specific data that can change the next generation, future generation people. And long read sequencing technological advances, developing long read sequencing technologies, as I mentioned earlier, such as those from Pacific Biosciences, that is PacBio from Thermo Fisher, that is what the advanced technology, and Oxford Nanopore technology will allow for more accurately assembly of complex genomics and better characterization of structural variations and repetitive regions. And for data analysis, machine learning, AI, artificial intelligence has been amended. So I think UK, US are just focusing on sequence and reporting all through artificial intelligence. AI has been making those things. So definitely give a particular, precise answer where we can change from the human errors. And in predicting disease risk, understanding complex genetic interactions, and optimizing treatment strategies have been precise informed through artificial intelligence. And then, as I mentioned, genetic counselors, clinical geneticists ultimately provide information. Yes, they have limitations whenever they got VUS things, but to or up to date knowledge and our data, what we can provide, we have to provide. And we have limitations, we can't ultimately explain the patients what they are getting through. And a lot of scenarios has been, as the previous case of Salvin, we know that it might be a de novo mutation. Sometimes we can't get into their shoes, and they have internal feelings, they can't able to detect their. And couple of days later, I have a special case presentation on the same thing, what we have get a case, and we have done clinical exome and then a whole exome, and then we are cross checking with different spectrum of testings like MRI and whole thing. We can't get enough data, enough particular reason behind that. So as such, there are a lot of cases we can't able to get a particular solution for what we are going through. So yes, that is what genetic counselors and clinical geneticists have a precise data. And we see in future these particular NGS will made an extraordinary work and get all the data, and will have a better solution for every case. And yes, the conclusion statement, this would be the breakthrough in genomic research and personalized medicine and diagnostics. And technologies continue to evolve and become more accessible. I hope this approaches to healthcare and scientific discovery, and then definitely transform the power of NGS in near future. Thank you. Thank you so much.
Thank you, Dr. Krishna. That was amazing. You took us from the different sequencing technologies to its applications to personalized medicine. We covered a lot today. And you answered so many questions that were coming up in my own head in the next few slides. So it was extremely comprehensive. We have a lot of questions in the chat box and comments for you. If you could please take them up. So the first one is, what's the depth considered for whole mitochondrial genome sequencing?
Oh, mitochondrial sequencing, that I'm not sure actually. Maybe I'll get through the answer and answer through the WhatsApp group. Maybe. Okay. Yeah, it's a good question. Right. Yeah.
Anyone from the audiences also, if they have any experience, they can please unmute themselves. Yes. And take it up. That wouldn't be a problem.
Yes, it would be helpful.
The next question for you is that many of the cancers don't have a single nucleotide mutation, but multiple mutations called passenger and driver mutations, yes, which might be identified during cancer genomic testing. How does NGS help in identifying these passenger or driver mutations, and how they play an important role in its management?
Oh, yeah, actually, I worked for 1.5 years in cancer clinics, cancer genomics, but majority of the things what they have gone through, the clinicians, even though to my knowledge, the clinicians and the patients are just focused on the NGS thing. Only they are get in hereditary cancers. So whenever they're surpassed with the somatic mutations, they never approach through the NGS, because all the biomarkers, all the chemical things that I have actually focused on to reveal the mutations. But whenever we are focusing on NGS, but they might need, but maybe I need to grow more about NGS, and I have to be, maybe I'm at beginner level to get more the details of things, but I'll definitely get through these things. And yeah.
No problem. We can keep the discussion on in the
Yes,
WhatsApp group as well. Definitely.
definitely. We'll get to know that. Maybe I'll try to get answers for that, and definitely we'll discuss in the group meanwhile.
Sorry to whoever's
Can I
having
add on to that answer?
Yes.
Yeah, sorry. So I'm from the NHS in the UK, and we have a big team here that does somatic work. And the importance of somatic work is directly relation to what therapy there is offered. So for example, if we see a V600E BRAF mutation in melanoma, or for in lung cancers or colon cancers, because all of these somatic mutations are tissue agnostic. So which means if it is a BRAF V600E mutation, you can, the treatment with any of the MEK inhibitors happens regardless of whether it is a lung cancer or melanoma. So currently the NHS guidelines works that way. So we test for the skin, at least melanoma testing, BRAF, NRAS, and KIT, because each of these have a different therapeutic outcome. Likewise, we test for fusions in case of lung cancers, the RNA fusions. So if they're positive for NTRK 1, 2, 3, then you immediately know that the patient is now eligible for NTRK inhibition therapies in chemo. So the big drive for cancer somatic mutation testing is directly to see whether we can put them on certain treatments, or more importantly, like if they have lung cancers and EGFR variant, so then, which means then they would not be eligible for a certain class of chemotherapy. Because the NHS here, since the healthcare is free of cost, so the important thing is to make sure that they have the right cancer burden to treat with specific treatment in somatic. So somatic is turning out to be a bigger and bigger chunk here in the NHS as against the inherited, because inherited of course has its own merits, but then somatic is now turning out to be more informative for therapy decisions. Thanks.
Yeah.
Thank you so much. Yeah. In the ocular setup, mostly what I get to see is retinoblastoma. We see ocular melanoma and a few other etiologies. So I have less exposure to other cancers. I am aware of these treatment modalities changing according to the diagnosis of somatic mutation profiles. Yeah, but thank you so much.
Hi, Ria. Hira here, sorry. I asked the question regarding mitochondrial genome sequencing depth. I only asked, but I think as far as I remember, it is 20x. I mean, correct me if I'm wrong, whoever's in the meeting, because I just wanted to make sure whether it's 20x that is universally considered for mitochondrial genome sequencing.
Okay.
Yeah, I wouldn't. I actually had that was my last question to Dr. Krishna also, that every now and then we have these reports of NGS where they'll give depth as 10x. And what do you say? Do we still, is it still important? Do we still consider it? In the sense that I will definitely, it urges me to order a Sanger for the same variant. But is 10x reliable enough, or what is the threshold cutoff?
Definitely there would be threshold of 30x. That is what a guideline says, because in individual sequencing, where we have a reads and then have been sequenced multiple times, the sequence might miss in the comprehensive database. And ultimately, that is what the guidelines mention, at least a 30x for the clinical exome, whole genome, and 100x for clinical exome, where we have a particular clinical features we are looking for, where we have a particular genes where we are looking for, at least they have to be sequenced 100 times then there might be a proper results, accuracy of precision result that can detect. So as you mentioned, definitely we have to go with the Sanger confirmation soon after NGS. We are doing for every case, irrespective of the condition and irrespective of the mode of the case. We do every soon after the NGS, we'll do the segregation analysis, I mean Sanger sequencing for the confirmation of the particular variant. Whenever it has been a significant conditions of a previous child with an abnormality or a family parents are carriers, then we'll definitely cross check with the Sanger. But the X depth is definitely essential. That's why we have to ask the person, NGS expert who are giving us the report. They might know that we are aware of the depth. That is what the point is. And to my knowledge, because I worked in a laboratory setups, and majority of companies, I can't able to say that majority of companies will malpractice that, because lower depth will give a lower cost.
Right.
So for that, they do a lower depth and they report it as 100x or 80x. So we can't at least conclude for the rare disorders. That is why we can't able to conclude, even though we picked for the particular panels and whole genome sequencing. So that is why the depth is essential. Yeah.
And from there on to Hira's question, do we know what is the threshold for a mitochondrial sequencing?
Yeah, she is saying that 20x, then
20.
yeah, we have to look into that. Might be.
Okay.
Because,
I'm guessing it's 15 to 20. I'm not too sure, but
okay.
this is something that I wanted to confirm.
Yeah.
Because generally you expect, at least 15 x depth in your mitochondrial report.
Yes, yes. Then I'll definitely check and get back to
Yeah,
you.
I think even I'm also researching, and I'll probably get back to you all on the group about
Sure,
that.
sure, sure.
Yeah, thank you.
Thank you.
Thanks. Yeah, that's all the questions, including the ones in the chat box. Thank you so much, Dr. Krishna and Dr. Saveleen, for today's session. And yes, we'll just connect again on the WhatsApp group for deciding what the next one's going to be about. Thank you everyone for joining in. Bye.